Hi, this is Neuroscience Technology Introduction. Before I get started, the major content of this lecture was prepared with a textbook by Matt Cart and Jennifer Shi, Guide to Research Techniques in Neuroscience. The course content mostly follows this order. Let me get started with a person who is regarded as the father of modern neuroscience. His name is called Santiago Ramon y Cajal. He was born 1852 until 1934. Of course, you can see he got Nobel Prize for his work. So how does he look? He was having a microscope, and he has, you can see various chemicals which we used to use Golgi staining method, and he was originally very good at drawing. So here's an example. So this is a preparation of from a spiral optic tectum with the Golgi technique. You can see these are beautiful single neurons and various kinds of neurons, which is drawn by Cajal. And his way of one eye looking at into this Golgi stained brain or nervous system, and then he draws this, and this observation and drawing remains to give a very important insight for the so-called neuron doctrine. So neuroscience is prepared by methods and new techniques. So quantum jump in neural knowledge is based on the techniques and technology. And here's a why, an example. So as we already say, Ramon Cajal, he was using this Golgi staining to visualize the morphology and the architectures of neurons and their circuits. And the second example is a famous Hodgkin and Huxley model for action potential, which is a membrane potential of when the neurons so-called fire, sending an electrical signal from one neuron to downstream another neuron, or here is what he uses as a big giant squid or lobster, and that has a big neuron. So they inserted an electrode inside the cell of the neuron. So what it is yes, intracellular recording to investigate the ionic basis of membrane potential. And those who have learned already the basics of neuroscience, this so-called firing is based on the ions, specific ions turn on and off, which makes these membrane voltages or potential spikes is propagating from up to downstream. So this is, yes, intracellular recording. And another very famous work in neuroscience is called Huber and Wieser. And so they use a cat to study a visual cortex. And what they actually did is, you see this backside of the brain, they insert a recording electrode. And so that what they get this as a signal is electrical signal, and that insertion of electrode is a near visual area of the brain. And what they did is they show cat a stimulus, a visual stimulus, which is lines moving left and right, or different angle and different angle. And then they realized different neurons will fire or respond to different angle of this. And so they investigate how information, especially visual information, is processed and recorded in the visual system. So what they did is insert the electrode not precisely inside the cells, but that's kind of outside of the cell. So it's recording called extracellular recording. So this new method gave a new ways of probing into the nervous system. And Ihar and Randy Shengman, they invented a so-called clamp technology to investigate the physiology of even single ion channels. Here we say ionic basis, and that is based on the ion channels present on the surface membrane of neurons. And you can see this a pipette. You clamp into this as a small list patch, which include a single ionic channel, so that by using this, they can measure the electrophysiological property of single ion channel. So that's why this method is termed as a patch clamp. Clamp is clamping, and they use a small patch, so it's called patch clamp. And have you noticed what the commonality of these new methods in neuroscience? Yes, they all got Nobel Prize, and these years are the years they got Nobel Prize. And more recently, Barack Obama, a recent president of US, has started a brain initiative. So still, there are so many things we do not understand, we do not know, an unexplored area in this brain. So new techniques and technologies are accelerating our understanding of our nervous system. So they started this brain initiative. So that is focusing on developing new techniques and technologies to accelerate our understanding. So how the brain stands for is brain research through advancing innovative neurotechnologies. So this initiative is really focusing on innovative neurotechnology development. So that's what Barack Obama said. As humans, we can identify galaxies light years away, and we can study particles smaller than an atom. But we still haven't unlocked the mystery of the 3 pounds or around 1.4 or 5 kilograms of matter that sits between our ears, which stands for our brain. So then what about these brain initiatives look for in terms of research areas? So this gives us a good idea what's actually necessary for studying the brain. So here, one is discovering diversity, identify and provide experimental access to different brain cell types. In fact, there are so many different kinds of brain cell types to determine their roles in health and disease. And we do not yet know all these different cell types' role and function in health and disease. And second, maps at multiple scales. So in the brain, we talk about circuitry. So every function seems to have a function and related to a circuit. So we want to generate circuit diagrams that vary in resolution from synapses to the whole brain. So synapse is the smallest junction which connects signal from one neuron to another neuron. So that is tiny, tiny, very, very small, even less than 1 micron size. So that to understand this, we can have very, very high resolution to looking into even single synapse or inside single synapse, what's happening, what are the proteins, what kind of ionic channels are there, how they function, there to the whole brain scale. So there are huge range of scales exist to understand this complicated brain. And 3 is the brain in action. Majority of our previous studies are studying a dead brain, which means a fixed brain, and we cut it off and then look it under the microscope, or we want to study the brain or nervous system most of the time, the tissue which is harvested. But now it's time that we have a better and more technologies which allows us to study the brain in its living condition. So we want to produce a dynamic picture of the functioning brain. Okay, fourth, demonstrating causality. So many of medical researches are we call correlative study, which means, hey, would let's say cell phone and brain cancer, maybe cell phone causes brain cancer. We can look at into the data that the number of cell phone increase may be linked to the increase in brain cancer. So these we call correlated, but correlation doesn't mean that it is causal. Cell phone is actually causing brain cancer increase. We still do not know. So causality is very important, especially when we are developing a new drugs or new treatment, because we have to focus on the real causal, the cause of this bad effect in resulting in disease. So we want to link brain activity to behavior with precise interventional tools that change neural circuit dynamics. So this way we can establish a causality, which is mostly regarded as important in scientific research into the brain. And fifth, identifying fundamental principles. Do we know all the principles in the brain? No. We want the conceptual foundation for understanding the biological basis of mental processes. Of course, we still do not know a lot about our mental processes. That's why we cannot control our emotion or our spirit very much. And on this research, this study probably will allow us to understand better about these biological basis of our mental process, including our thinking. Okay, 6, advancing human neuroscience. A lot of these studies are done in a lower level organism or mice, some rodents, but many people would argue in the end we care about to understand our brain. So that's why human neuroscience is important. We want to develop innovative technologies to understand the human brain and treat its disorders. So in the end, our ultimate goal is more likely to apply these knowledges into understanding our human brain and human brain diseases. And finally, we integrate new technological and conceptual approaches. So let me start with a scientific hypothesis. So what is scientific hypothesis? So what is hypothesis? Hypothesis in a simple way is a worldview that is testable, okay? So what is scientific hypothesis? Which is an idea that proposes a tentative explanation or educated guess about a phenomenon we observe. So the formulation and testing of a hypothesis is a part of scientific method. So there are two important qualities of a scientific hypothesis. First, as I say, a worldview that is testable, which means we test and see if it's true or not. That is the core part. So scientific hypothesis must be, yes, testable. And so what it means is we can develop an experiment that could be used to test the validity of the statement or the hypothesis. And the other is that experiment could be devised that might reveal that the original hypothesis is not true. So that experiment must be testable, and the result could be, yes, we accept the hypothesis, or it turns out that the hypothesis is wrong. So this aspect is called falsifiable. So these two qualities should be met. Otherwise, we are not talking about scientific method. So I want to give an example of my own. In fact, from my previous work, let me explain. Improving treatment of brain metastasis. So metastasis is a cancer growing, spreading in another organ. So for example, breast cancer cells can got into the brain, and we call brain metastasis. We want to improve the treatment from breast cancer, which cells are so-called HER2 amplified. What is HER2? HER2 is a kind of growth factor which makes the cancer cell to grow fast. So in this case, human epidermal growth factor receptor 2. So what's my hypothesis? So my hypothesis is to better treat these tumor cancer growing in the brain, not in the breast. So tumor microenvironment contributes to differential response, different response to HER2 inhibitor, which is a treatment which blocks HER2 antibody, HER2 signaling in primary breast tumor versus brain metastasis. So this hypothesis is, hey, the response of treatment, even the same treatment, to the tumor growing in the breast or brain could be different, and that could be due to the different microenvironment. So this is one example. So this control is a vehicle, and we have HER2 anti-HER2 antibody, and we have another drug which blocks this HER2 signaling, small molecule drug. And so look at this. This x-axis is a time, y-axis is a tumor value. So in the mammary peptide or a model of breast tumor growing in the breast of the mouse, so you can see the tumors are growing over time, but with these specific treatment, the tumors are not growing or controlled, which is good news. However, the same tumor cells are implanted into brain, so-called mimicking as a brain metastasis model. You see what happens over time. It exponentially grows with the vehicle, but even with this treatment, they are not controlled, but they have some delay, a little slowly, but they eventually keep growing. So what are the differences between these? The same cells growing either breast or brain? Yes, that locations makes a difference, so that we call it as a tumor microenvironment. Of course, after this, we better to understand what's happening there. So this is kind of an example of an hypothesis. Another example I want to bring it is complicated hypothesis with a large data. So for example, we can have a hypothesis, hey, drinking alcohol gets you live shorter, you die earlier. A good hypothesis. How to prove it? Then we better to have a data set. In this case, you may gather a huge data set. In this case, almost 600,000 drinkers and a lot of studies, and so that we can find out and more specific information, so-called risk thresholds for alcohol consumption. How much alcohol drink would be, not affecting my lifespan. So here's a very interesting result. You can see, let's see first this cardiovascular disease. Hey, x-axis is a gram of alcohol per week. Oh, look at this. Maybe 100 grams per week is in fact a lowering cardiovascular risk. However, you can see it's a higher when it goes more and more drinking. By the way, the y-axis called hazard ratio. Hazard ratio of 1 means this risk of dying is the same as control group. So that if this is 1.5, which means 50% higher chance of dying. So all causality, cause mortality, you can see the alcohol consumption as the same as almost 1, and you can see it start to jumping for 200 grams per week or 350 grams per week. And then you can lose your years of life with much higher alcohol consumption as your ages. So in conclusion, in current drinkers of alcohol and mostly high-income countries, the threshold for lowest risk of all-cause mortality, you can see, is about 100 grams per week. Maybe 100 grams of alcohol per week, maybe is allowable, okay? But to draw these conclusion, these are 95% confidence interval, and these requires a various statistical analysis and interpretation. So studying statistics or biostatistics is also very important.